Yeah, look, I do want to say thank you very much for coming. And I think also as a scientist, as a medical researcher who lives in this beautiful, edifice to medical research, it's really a pleasure to have the opportunity to directly tell, non-scientists about what we do. So it is really, I really thank you for coming and giving me the opportunity to talk about our work here at Garvan. So we're going to talk about, I'm going to give you a little story about type 1 diabetes and what that really is. It's a disease that children get, but then once you have it for your whole life. And to tell you about type 1 diabetes, I need to tell you a little bit about how sugar in your body is controlled. I need to tell you a little bit about your immune system. And then I'm going to bring those two together and tell you a little bit about the disease, what we're doing here at Garvan to help prevent it and stop it, and some of the exciting sort of developments both in the basic research and in what's happening in the clinic. And again, from the perspective of what the Garvan's doing. Okay, so buttons. So sugar, how is sugar regulated? So you have to sort of zoom in to your body, open up your abdomen, and you'll find this sort of squishy thing called the pancreas. And it sort of runs along in here, along your intestines. And there's like another cartoon of your intestine there, and there's this sort of pancreas here. I'm sort of cut it long ways. And you can see one of the most clear features is this huge sort of tubey thing. It's a pancreatic duct. And one of the things the pancreas does, it's full of all these tissues that release digestive enzymes that channel along this duct into your intestines to digest the food that you've had for brekkie, right? But there's another sort of trick to the pancreas, another secret that wasn't actually discovered till the 1850s. And this is a tissue biopsy from a real human pancreas. And what's happened here is we've added a chemical that will stain most of the pancreas blue. You can see that's not naturally a blue color, but you can see that pancreatic duct here. And all these little blue blobs here are the enzyme, or the clusters of the cells that secrete the enzymes that digest your food. But you'll see there's something else kind of different about the pancreas. There's a sort of blobby brown thing. And this was noticed by a German medical student in the 1850s named Paul Langerhans. So we kind of named it after him. So he's got that honor for spotting it first. And I guess it's like a spotter's fee, right? That's how it works in science. So we call these the Islets of Langerhans, or islands in English. And what we've done here is added another dye. And this dye picks up the hormone insulin, and you probably know about insulin. And you can see that this structure is really particular because it has lots of the brown stuff, and the brown stuff's nowhere else. So these islets of Langerhans are really quite cool. They're a beautiful sensor in your pancreas that can actually measure second by second how much sugar is in your blood. And when the sugar levels go high because you've just eaten and food is now coming in the blood from your tummy, the islet cells will release this hormone insulin into the blood, and it travels with the sugar to the liver, and it tells the liver, hey, there's a lot of sugar coming through. We've got to get the body to start storing it to bring sugar levels down. And this is really important because sugar is literally the fuel for your body in the same way that petrol is the fuel for your car. I guess if your car runs out of fuel, you sort of pull up at the side of the road and call the NRMA and everything's kind of sorted. But if you run out of fuel in your body, that is not sustainable, that's lethal. And so it's really important that sugar levels are kept within very defined ranges. And that's what these pancreatic islets do. So, okay, so now we're going to have a little journey in the immune system. I'm going to steal a couple of those high-resolution photographs that Rob had, that his lab is expert in. I'm going to play them on you now. And so we know that the body has got this blood vessel system, and that's the highway of your body and the pathways by which the immune system uses to get around your body. And look, I'm a sci-fi fan, I have to admit that. I don't know if any of you remember this movie, Fantastic Voyage. I just love the idea, the spaceship thing, and they all got shrunk and injected into the blood. Remember, and they were pulled out of the eyeball at the end. It was really cool. So I always sort of imagine if you could get into a spaceship like that, or a little, some sort of weird submarine and be shrunk, what would it look out the front window when you're running through the bloodstream, right? And again, am I really loud?
No, it's feeding back, that's it.
I had a really big coffee this morning. Is that better? You can turn down your whatever. How's that? Is that better? You like that better? Can you hear me now? So if you look out the front, look, you've seen this. Rob was telling you this is what the bloodstream would look like in an illustration, but what's kind of cool about this, it tells you a lot about immunologists, right? So the red stuff, what do we call that? Red blood cells, right? The white stuff, there you go. And you know how we describe these? Go and have a guess. donut-shaped. You get it. You can see where immunologists are going. We're obviously a pretty crude lot, right? But the immune system is really important, as Rob said, right? And I think what he made a comment at the start that we need the immune system, we have to have it. But then he said sometimes it misbehaves. And medical research is about how does it work normally, that's really important, but also what's going on when it misbehaves. And I thought we're going to talk about how it misbehaves, but it's also important to show you some of the cool things it does. And like the immune system is anti-cancer, and this photo is a little bit low res for the big screen, but these white blobs, these are now the white blood cells, they're the immune system cells. And this here, plated out, sort of stuck to this surface, is a cancer cell. And these immune cells are attaching to it. And see how this one is kind of elongated? They do that when they're angry and activated. And these are actually dropping digestive enzymes and nasty free radicals and other chemicals onto the cancer cell to kill it. So that's one of the key functions of the immune system, is an anti-cancer defense system. The other thing that the immune system does is it eats bacteria that enter your body. This is cool. This is a real cell. It's called a macrophage. Sounds fancy, but again, going back to simplicity of the immune guys, macro means big, because it's a big cell, and phage means eat. It's a big eater. So again, you can sort of see how immunology sort of progresses. But this is in green here. This is a bacteria in all seriousness. And you can see that this guy's actually reached out this kind of weird pseudopod, and it's actually going to grab that bacteria, pull it inside itself, and dissolve it. Right? It's pretty cool, right? So the immune system has all these incredible functions. This is real-life image of an immune cell in green chasing a bacteria in red. It's from a colleague of mine, Tatiana Chatnova. But this is like really in real time how immune cells look. This is like a giant Pac-Man. Check it out. Chasing a real bacteria. Look, the bacteria is trying to get away. The immune system is chasing it. Poof, gone. And it's really amazing, right? I mean, immunology is fantastic. So that's when it's good, right? What about when it's misbehaving, as Rob said? And that's a really nice, elegant way to put it. So this is another sample, another biopsy from a patient. This time it's a person who has type 1 diabetes. And again, this is from the pancreas. There's going to be a test at the end of this morning session, isn't that right, Rob? So I'm assuming now your pathology is up to scratch. So you can see immediately this is a pancreas biopsy. You can probably recognize the ducts here. But these are the cells that secrete the digestive enzymes. But there's your pancreatic islet, right? You can sort of pick that out. Looks pretty normal. This time we've used a different dye to highlight the tissues. It's pink, right? But what you see here is another pancreatic islet, but it looks really different, doesn't it? All those small dots, each one of them is actually a white blood cell. And we know they shouldn't be there. They've actually come there to destroy the cells that make insulin. If you put the brown stain on again to look for insulin, which we did in this biopsy, you don't see any brown, it's gone. So the cells that make insulin have been absolutely destroyed by the white blood cells. And here's another picture down here. We've added the stain. The brown here is not picking up insulin, it's actually picking up very specific types of white blood cells, in this case the T lymphocytes or the T cells. And you can see these T cells here surrounding this pancreatic islet. So what is type 1 diabetes then? This is the sort of the fast facts, the take-home message. And this is again another image, another really beautiful colour image. This time we've got a fluorescent dye that's red that picks up insulin, so you can see your nice round pancreatic islets. And the green here are immune cells. These are T cells, and you can see they're all swarming around wanting to attack. So what is type 1 diabetes? Well, it's not the same as type 2 diabetes. They're very different. Type 1 diabetes is about an autoimmune attack. Your immune system attacks yourself and destroys those cells that make insulin. Once they're destroyed, meaning absolutely killed and gone, no other cell in your body or other tissue can make insulin. And so you can't control your blood sugar anymore. And your blood sugar can go crazily high and crazily low. And if it wasn't for the fact that we have insulin, it would be lethal. Right? That's the brute fact about it. But and it affects about 122,000 Australians currently. So why do we get it, and what's it all about? And that's what we're here. One of the things we're trying to work out here at Garvan. Well, we know now that it's a genetic disease, right? It runs in families. You can also get it de novo, but there's about 20 genes involved. It's a kind of a curious disease because something's going on with the environment as well. It's not just genetic. There's an environment and genetic interaction, and that seems to be the case for a lot of sort of diseases that we get, these very complex interactions with us and the environment. But why do you get this? Why does it actually start? In a sense, what is the trigger? Like if you have the genetics and maybe the environment, is there something that precipitates it? And we still really don't know that, and that's probably the biggest question in the field. Because obviously if you knew that, you could try to prevent that from happening, or people have been exposed to whatever that trigger was. So one thing we've been trying to do in Australia, what we have done in Australia, is to work out ways to prevent it but also treat it. And look, you do know if you've got colleagues or friends or family with type 1 diabetes, you know they take insulin, right? They take it with a needle and they measure their blood sugar. And that's fantastic. But there's a small percentage of people who for that is not really good enough. And they develop what we call diabetic complications. They get damage to their kidneys, they get damage to their eyes. They can also have a thing called hypoglycemic unawareness. It's a very technical term, but it means they can't tell when their sugars go too low. And they can fall unconscious. They can't work, they can't drive. It's a very disabling disease or very severe complication of type 1 diabetes. So for these people, current insulin therapy is not working. And so for this reason, we were thinking, well, again, fairly simply, if the immune system has destroyed your cells that make insulin, why not just put new ones back? And we know that that works with, you can lose a heart and a heart can be put back and it works in a transplant model. So what we thought is, why don't we do islet cell transplantation? Take islet cells from organ donors and put them back into patients. And so the federal government set up this thing called the Australian Islet Transplant Consortium, which is actually now a national funded centre. And it had 3 sites, one at Westmead, which is kind of the lead site of which the Garvan was a part of, one in Melbourne and one in Adelaide. And I thought I'd just show you, so it's a pretty gory business transplantation, of course, but this is a bag which actually contains real live human islet cells that have been taken from an organ donor who donated their pancreas. And the islet cells are prepared into a solution, and then a small incision is made in the abdomen, a central line is put in, and the islets are actually infused into the portal vein, and there's all this sort of cool technology to track that the islets are going into the liver in the right spot. But this is what it looks like for a person who has type 1 diabetes and then gets an islet transplant. So the 2 key things, this is actually a clinical chart. Here is your blood glucose levels of this person. And the other really important thing really is this is how much insulin they were taking each day. Right? So basically you can see their sugar levels go from very low to very high, really not very well controlled. And this is a small group of people with type 1 for which this treatment is designed. They get an islet transplant, TX1, bang, and you can see immediately already the sugars are really looking much better. They're coming down and they don't have so many big highs and lows like you're seeing here. And this is what we're going to talk about. For islet transplantation, it seems like you need a second transplant for it to work. It's like saying going to get a kidney transplant and then a year later you've got to go get a second one to help the first one work. Right, so the technology is really amazing, but it's not working as well as we want. And this is one of the big research questions that we're interested in. But what's really cool is when this person gets the second transplant, you can see everything sort of looks a little bit better again. But look down here, this is the really cool story, right? The person goes off taking insulin, and this is like 4 years ago. So this is like one of the success stories of this transplant program. So what's the problem? The problem is, if you look at, there's your pancreatic islet transplanted now in a mouse in an experimental model. And these are these green cells are the immune cells. And you can see as soon as we do the transplant, these immune cells are all over the transplant. They're sort of crawling all over it, like sort of bugs, right? And the issue is that we need to be able to deal with that. What are the interactions there? And if we could manipulate that in different ways, could we reduce the need to have a second transplant, and could we improve the success of organ transplantation in general, but also islet transplantation? So I'm going to take a chance here, assuming that you're quite sophisticated because you come to the Garvan sort of things a lot, and actually show you some real scientific data. So this is just one snapshot, and I'll try to keep it simple, but wave or yell at me or something if it doesn't make sense. But what I really want to show is how we're trying to manipulate the immune system and the sort of changes that we can get in the experimental models that I think in the future are going to lead to really big breakthroughs in the clinic. So in this case, this is what we call a Kaplan-Meier survival curve. And basically all we're looking here is we give transplants to mice and we ask, how long do the transplants survive? So if you give a transplant to a mouse, they all get rejected. That's normal. You have to give lots of immunosuppression for a transplant to survive. But what we've been doing here is we've been looking at the interactions. Remember the crawly bugs all over the islet? We've been adding anti-inflammatory compounds to the graft to see if we can change that environment. And when we do that, we get this sort of dotted line. So you can see the grafts do much better, right? We actually have about half the animals survive, and we don't give any immunosuppression. But in the clinic, immunosuppression is really important. And we thought that one of the problems with immunosuppression, though, is it has a lot of side effects. But if you could reduce the immunosuppression, you might have the benefit without the side effects. So what we did is we went down to our local health food shop and dosed out immunosuppression into what we call a homeopathic level. So that's the green line here. So we add immunosuppression to the recipient mice, and you can see it doesn't have any effect on the transplant. When you put this new anti-inflammatory drug with the low-level immunosuppression, you get even better outcomes. And I think this is the sort of thing, this is the sort of breakthrough research that's happening at Garvan, where you can imagine what the future could look like and how that would meld in with current best practice to actually improve it even further. Look, these are the guys who actually do the work in the lab. And look, I really thank you once again for your time, and it's been a privilege to tell you about our research.